Okay, so in the next slide we see the connection between G and C, A and T, and A and U, and you can see that the upper bond is a very stable bond, which elevates Tn because it has three connections in the middle. You can see the less stable bonds between A and T and A and U, which will decrease Tn. It means that the more GCs that you have, the more stable the molecule, the higher Tm is. The more AT/AU bonds that you have, the less stable the molecule, the smaller or lower the Tm is going to be. So under the right conditions, we can renaturate DNA and RNA exactly like we separated them. We can connect them, and the process is called renaturation. So basically, renaturation is the opposite process to denaturation or melting. Okay, melting, denaturation, separating the strands, renaturation, connecting them together. In addition, we also call or term renaturation as annealing or hybridization. So you might find those two terms as well. The strands are not perfect. If the strands are not perfectly matched, we will have an imperfect duplex that will be produced in the hybridization process, right? Okay, so now we're going to address mutations. Mutations are called mistakes made along the way of the process from DNA to protein. So we have two phases in the middle, right? Generally, transcription first, going from DNA to mRNA, which we will talk about, and translation from mRNA to protein. So any mistakes made in the way are what we call mutations. And I would, we're using the word mistakes, but we'll talk later about are these truly mistakes or not. So you can understand that the answer doesn't have to be yes. And we talked about mutations in the first presentation. Mutation can give a certain organism an advantage that it didn't have before, and then it's usually conserved. It can give it a disadvantage, and then it's usually deleted, although not always, or a mutation will not affect either way. So it's there, but it has no meaning, no true meaning, and then it will also not be observed in the naked eye or without really going into the gene itself and finding it. Okay. So mutations that are mistakes were and still are a key to understanding of sequences of DNA, protein subunits, and function. Because here's the thing, let's say that you have a gene that is perfectly functional right now, and a mutation, a mistake, occurs, and now the function disappears. It's no longer able to function. So if you can find that mutation, and if you know what mutation caused that, you know exactly what part of a gene or what the gene itself does. With that mutation, something does not occur. Or, for instance, you can have a mutation that makes you have the ability to do something. So without the mutation, you wouldn't have the ability. So that is a very important part of understanding the role between genes and functions, because mutations usually change something. And when something changed, you understand how that mutation contributed to that. Mutations are rare relatively to the number of processes that is going on in a human being, but they also may be deadly. Okay. Right now, till date, there are more negative mutations than positive ones. So as of now, through evolution, mutations usually mean that we're going to have a disadvantage or a disease produced. And it might also mean that at this point of time, in a way, and I'm being very cautious, evolution has reached a certain end point where there's no more advantages to be earned. But I'm saying that cautiously because we are going to discuss evolution, and what I just said might be a mistake, and I'll explain it later on. Okay. Some mutations are just spontaneous, meaning that something's going on in the cell or in the organism, and a mistake just happens in that specific organism, and it's just spontaneous. And it just happens through normal function or metabolism. It doesn't mean that that's happening at the same time to every organism or every cell. Okay, so it can also be a local mistake. But the existence of mutations throughout generations means that it has gone through, and this is a very important term that I need you to understand and remember, what we call selective forces, meaning these mutations, good or bad, that are conserved throughout generations are actually competing in a way against evolution. Okay? The chances of something being conserved are not equal to, are not the same for every mutation, and they are actually working against, so to speak, evolution to be conserved. Right? Because once again, you are conserving, so to speak, a mistake, which the system generally and naturally is against. A mutation can be induced, so that will be called an induced mutations, and the factors that are generating or inducing those mutations are called mutagenes. So for instance, if there's some kind of substance in food or in the air, if you smoke, certain drugs, those all can be mutagenes that might create a mutation, good or bad, meaning giving you an advantage or disadvantage. But once more, till date, there are more negative mutations than positive, right? So we have a mutation, all right? Let's say that we have a mutation that causes people to have two noses. Now we can also calculate what we call the background level, meaning what is the ratio of occurrence of that mutation. If half of the population would have two noses, that means that that mutation has a high level of occurrence, meaning its background level is very high. But even if only 1 in a billion has it, it means that it's low. Okay? The higher your background level is, the more likely for the phenomenon to be found or be observed in a certain population. Okay? So the use of mutation generating processes can actually be used by humans and has been used by humanity and will be used in the future to produce a certain outcome. Not so long ago, I read an article on the internet about a scientist that decided to grow an ear on one of his hands. Okay, so what he actually did has created a mutation, right? Because how many of us have ears on their hands? Don't answer that. The mutation police will come to look for you. The ability to change a gene exists, and there are many different mutations that can be produced naturally or artificially, right? So we have that option. In a molecule, as long as DNA, there are so many different combinations of mutations that are possible. Okay, so let's say, let's take your phone number. Your phone number has 10 digits, right? 10 numbers in it. How many different combinations of mistakes can you make in your phone number? Let's say that your phone number, for example, is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. You can switch between the 1 and the 2. You can switch between the 1 and the 3, and so on and so forth. If you think about all the combinations of mistakes that can be made, you'll have a very big number, right? But here's the thing. If every number represents a nucleotide and DNA has up to millions of them, what happens now to all those combinations of mistakes possible? It's almost endless, right? So there are so many options of mistakes that might be made. In the introduction video, we talked about point mutations, and point mutations can be caused from a change in the parental DNA or during the replication process. So point mutations can be basically produced almost at any time. For instance, let's say that instead of an A, we now put a G in the sequence. For many reasons, guanine has just replaced an adenine in the sequence. That also means that instead of a T, we're going to have a C connected, right? Because the base pairing says that A connects with T, G connects with C, and vice versa. Those are the two options, right? Such a point mutation is called transition. So we had a transition from the A base to the G, or from the T to the C. And once one of those four options occurs, immediately the base pairing changes, right, accordingly, because we're working with pairs. If we have a switch within the same base, within the same pair, meaning that if the A is right now here, and the T or the U is here, and we switch them, now the T is up here, and the A is down here. That is what we call a transversion. Okay? Once again, transition is a change in one of the bases that changes automatically its pairing, right? Transversion is kind of flipping the pair. Okay, we'll stop here and continue in the next video.